I'm a hematologist with a special interest in myeloproliferative neoplasms, particularly myelofibrosis. So myelofibrosis is an advanced myeloproliferative neoplasm characterized by fibrosis in the marrow and extramedullary hematopoiesis with massive splenomegaly. Patients with myelofibrosis do experience debilitating symptoms related to the disease itself and also due to massive splenomegaly. The current standard of care for patients with myelofibrosis is treatment with drugs called JAK inhibitors. There are two agents that are currently approved for treatment of myelofibrosis that are JAK inhibitors. One is a drug called ruxolitinib, and the other drug is called fedratinib. So these agents are very effective in treatment of myelofibrosis. They improve the quality of life, decrease the spleen size, and also alleviate many of the symptoms associated with myelofibrosis. However, a number of patients have inadequate response to these drugs and also develop progressive increase in spleen size after an initial response. At present, there are no approved agents for treatment of myelofibrosis, and there are a number of investigational agents for patients that fail JAK inhibitors. A few years ago, our group at Huntsman Cancer Hospital, University of Utah, published a paper in clinical cancer research. That was actually my fellowship research project. So we did a library screen utilizing a short hairpin RNA on an MPN cell line. And we came across this protein called RAN as one of the top hits in our genetic screen. And RAN is involved with another protein called XPO1, and together they are responsible for nuclear cytoplasmic transport, transporting tumor suppressor proteins outside of the nucleus. So when we found this gene, we further investigated a pharmacological agent called selinexor, which blocks this protein XPO1. So selinexor is an oral small molecule inhibitor of XPO1 and has shown broad activity in a variety of cancers. It is a drug that's currently approved in the United States for treatment of multiple myeloma and also diffuse large B-cell lymphoma. So we further did preclinical studies and found that JAK2 positive cell lines are very sensitive to inhibition by selinexor. And we also showed that selinexor preferentially or selectively, I should say, decreased the proliferation and increased the cell killing of cells derived from myelofibrosis patients compared to normal cord blood cells. So based on this preclinical data, we conducted a study to evaluate the safety and efficacy of selinexor in myelofibrosis patients that were either refractory or not tolerant or intolerant to JAK inhibitors. So this was a single center investigator-initiated study. We initially designed the study to enroll 24 patients. It's a prospective open-label single-arm study. And the patient populations are patients with the diagnosis of either primary myelofibrosis or post-PV or post-ET myelofibrosis. All these patients should be either refractory or intolerant to JAK inhibitors. We defined refractoriness to JAK inhibitors as persistent splenomegaly and symptoms of myelofibrosis after at least 3 months of JAK inhibitor. And then we also defined intolerance to JAK inhibitors as any side effects that led to discontinuation of these drugs. And these patients were treated with oral selinexor at a dose of 60 to 80 milligrams once a week as a starting dose. And the drug was continued until progression. And we evaluated a response to the oral once a week selinexor by utilizing MRI scans to estimate the spleen volume. So the primary endpoint of this study is spleen volume response rate, which is basically defined as more than or equal to 35% reduction in spleen volume from screening to week 24 MRI. And we also collected some symptom scores to measure the total symptom burden of these patients, and also obtained a bone marrow evaluation both at screening and also at week 24 to see any changes in the bone marrow fibrosis grade. We've treated 12 patients so far in this study. 7 were men and 5 were women. And median age was 68 years. And median duration of prior JAK inhibitor therapy was 22 months, which is quite long and does indicate that these patients were prior treated with JAK inhibitors for an extended period of time. And 11 out of 12 patients were considered resistant to the prior JAK inhibitor therapy. And many of these patients, I think 8 out of 12 patients had at least one high-risk molecular mutations. And what we observed was that just single-agent oral weekly selinexor induced very impressive rates of spleen response in these patients that were previously refractory to JAK inhibitors. 30% of the patients had a spleen volume response rate of more than 35% at week 24, and 40% of the patients had a spleen volume response rate of more than 35% at any time during the study. And 60% of the patient had at least 25% reduction in spleen volume on the study. And the other interesting finding is that we also observed improvement in anemia in some of these patients. For example, 4 out of 8 patients who had hemoglobin of less than 10 grams at screening had an improvement in hemoglobin. And more interestingly is that one of 2 patients who had transfusion-dependent anemia, 1 patient became transfusion-independent and remained without requiring transfusions for almost 2 years on selinexor. So these were very impressive findings, particularly that outside of the JAK inhibitors, a single agent demonstrating responses both in terms of spleen and also in terms of improvement in hemoglobin. The other key finding that I would say is that we've noticed, we observed a reduction in the white blood cell count and LDH, which reflect the underlying myeloproliferative activity very quickly after starting selinexor. And these 2 parameters remained under control during the whole time on selinexor treatment. In terms of the side effects, we did observe weight loss as the most common side effect. Majority of the patients were able to tolerate with dose adjustment, and this was the weight loss was manageable. However, 1 patient had to stop the treatment because of the weight loss. And we also observed fatigue and dizziness in some of the patients. And, 10 patients of the 12 required dose reduction because of the side effects. But I think in terms of the side effects leading to discontinuation of the drug, we had 3 patients who stopped the drug because of side effects, 1 due to fatigue and 2 due to weight loss. So overall, the patients remained on selinexor for a good period of time. The median treatment duration of selinexor in these patients was 11 months. And there were 3 patients who were able to continue the drug beyond 18 months. So, this indicate that despite some of these side effects, that patients were able to stay on the treatment for an extended period of time. And more importantly, these side effects, particularly fatigue and nausea and dizziness, improved over time on treatment. So, and we haven't really observed any sort of serious organ-related side effects on the treatment. The other interesting finding is that we examined paired bone marrow samples at screening and at week 24 in 10 patients. we didn't see any change in the grade of reticular fibrosis. However, we had one patient who had a bone marrow evaluation done at week 22, which is, almost 1.5 years after starting treatment with selinexor. And this patient, we observed a reduction in the grade of reticular fibrosis from grade 3 to grade 1. So this does indicate that the selinexor has a potential disease modification effect on on myelofibrosis, and perhaps we need an extended treatment duration to observe these changes in the bone marrow. So we're planning to collect additional bone marrow evaluations from some of the patients who are still on selinexor beyond 18 months. In myelofibrosis, after failure of JAK inhibitors, there is a huge unmet need. We have a number of agents that are currently in development. And some of those are, for example, navitoclax, which is a BCL2 inhibitor, has some promising data. And there's another drug called pelabresib, also has a promising data. But I strongly believe that selinexor has shown a single agent activity which is outside of the JAK inhibitor class of drugs. And we are trying to determine the right population that would benefit from selinexor, either upfront or after failure of JAK inhibitors. So there are 2 multi-center, multinational studies, registration studies are currently ongoing. We are also participating in those studies. One of the studies is looking into combining selinexor with ruxolitinib in newly diagnosed patients that are not previously treated with JAK inhibitors. And we're also doing another study in patients who are previously treated with JAK inhibitors and were either refractory or intolerant, and get randomized to either selinexor versus physician choice. I think these studies hopefully will accrue patients faster, and we can determine the efficacy of selinexor in a randomized fashion in both JAK2 refractory and also a JAK2 naive setting in combination with JAK inhibitors. Myelofibrosis has been a difficult disease. The only cure at present is an allogeneic stem cell transplant, but many patients don't qualify for a transplant, either due to age or comorbidities. But also there are a number of long-term complications of allotransplant, particularly chronic graft-versus-host disease. So it's really important to find effective treatments for patients with myelofibrosis that can add to the drugs that we already have for these patients. And my hope is that we will find effective treatments that could potentially transform the landscape of myelofibrosis treatment and make this disease into more a chronic illness, even if it's not curable, with improved quality of life and also an extended survival for these patients.